Hello, everybody, and welcome to today's NCBI webinar. We're going to talk about the Genome Data Viewer today. This slide just has some preliminary information that you may need. Wayne Madden is here with me, and he's going to answer questions during the webinar while they come up. All the questions and answers are going to be available after the webinar in a document that's going to be linked to our webinars and courses page. It's also going to be available in our FTP directory for this particular webinar, and that's the materials directory. There's a compressed URL there that will take you to that directory. Okay, so let's go ahead and get started. I'll show you a few slides, and then we're going to go over and do a live demo for the rest of the time. The Genome Data Viewer is the topic for today, and mostly today I'm going to say GDV when I mean the Genome Data Viewer, just because it's a little bit faster. And again, my name is Peter Cooper. As I mentioned, if you have any questions for me, you can write to me at that address at the bottom of the slide, petercooper@nih.gov. We're going to talk what the Genome Data Viewer is. We're going to explain that a little bit. We're going to talk about accessing it, a brief overview of the interface and functions, and mostly what we're going to do is demonstrate it live for you today. So the Genome Data Viewer is now NCBI's main genome browser, and that really means the assembly browser for eukaryotic organisms. And this is replacing the Map Viewer, which is our original genome browser. You'll recognize the interface. It's similar to the other specialized browsers that we've had available at NCBI, like the Thousands Genomes browser and the Variation Viewer. Embedded in the center of all of those browsers is the familiar, I hope to you, Sequence Viewer panel that's present in places like Gene and in Nucleotide when you do the graphics view. It has functions for navigating across the assembly, uploading map data, accessing analysis tools. And right now in the Genome Data Viewer, we have over 500 eukaryotic genome assemblies available. So how do you access it? There is a home page that you can easily get to. Probably the quickest way to find it is to search it in Google. It has, by default, you're going to search the human genome. You can also use this phylogenetic tree function that's available there to sort of browse the various kinds of organisms that you can load into the Genome Data Viewer, their assemblies. So for example, I can expand the node there that contains zebrafish plus a bunch of other bony fishes, and it's a convenient way to navigate and browse by organism. Today we're going to be focusing on a particular assembly, actually more than one, for a particular organism, and that's the human genome. You can search with gene names in this box here, SNP IDs, RefSeq accessions, chromosome positions. You can pick the assembly you want, the current genome assembly, the current reference assembly, GRCH38, or the previous assembly, GRCH37. You can also search some alternate assemblies, the HuRef assembly, the Celera genome, and the identiform molar cell line assembly, CHM1_1.1. There's a browser function where you can enter the browser itself at chromosome 1. You can blast a specific database that contains the particular assembly you're interested in. And of course, this ideogram view allows you to click and browse a particular human chromosome if you like. Another way to get to GDV, and these have been around for a while actually, in various places at NCBI. So from Gene, I can link to the Genome Data Viewer. In assembly, I can link to browse the assembly in Genome Data Viewer, for example, the dog assembly in this particular case. And today, as a matter of fact, we now have links from BLAST to the Genome Data Viewer, and that went live this morning. So here's the main browser interface, and like I said, it should look familiar to you. It's sort of in the middle of it is the graphical sequence viewer. And then around the outside, on the left-hand side, at the top, are various search and navigation features. If you run a search, you can get your search results there in the ideogram view, and it shows you the hits to the various chromosomes. If I search for a gene name, I might find hits in more than one place because it's sort of a text search. I can select the one I want from the panel right underneath that. Then there are widgets for uploading data, adding tracks, loading BLAST results, and we will use those in a few moments when we do the demonstration. At the top, there are some navigation functions as well. There's a gene and exon navigator that's present in our other browsers, and we'll take advantage of that. And at the very top, there is an ideogram with a particular chromosome that you're on, viewing at the moment, in this case, chromosome 1, and I can use that as a way to navigate and display various parts of that chromosome. So just a little detail on some of the additional functions, and we're going to use these today. Hopefully we'll get a chance to use all of them. I can upload my own data. And these are the various kinds of file types. It'll take BED, GFF3, GTF, GVF, BCF, and HGVS. I can also, if the data is present on a web page, I can use an URL, or I can simply paste the data in the text box there. I can run BLAST from within the browser and load the alignments there. And there's a little tool called the Alignment Inspector that lets me sort of navigate across my BLAST matches using either the query coordinate system or the genome coordinate system. I can look at assembly region details. So there are places where we have alternate assemblies for the human genome because you can't represent the diversity of the human genome in one particular assembly. So we have ways of browsing those. And then finally, there are some NCBI data sets that are mapped to the assembly, and you can load those through the add tracks function. And of course, you can add all the tracks that are available in the graphical sequence viewer. Let's go ahead and do some live examples. I'm going to focus on 2 chromosomes and a couple of different things on one of them and one sort of area on another one. So the first thing we're going to do is look at the region surrounding the adenosine deaminase gene on chromosome 20. We're going to add some of the recommended track sets that NCBI has. We're going to configure the tracks that are there. We're going to upload some custom data. I'll show you how to print and share views from that. And then from that particular gene, we're going to run primer blast to get some primers to amplify a particular exon. And then we'll blast some sequences from different species, and we'll load those into the browser to look at conservation. And then on chromosome 2, we will, if we get a chance, we'll take a look at region 116, which is a region that has alternate assemblies. And then we'll look at the region surrounding the lactase gene. And when we look at the lactase gene, we'll look at gene expression tracks that are available, as well as one that I made myself from the tracks that are available in the graphical sequence viewer that has to do with exon coverage. And then we'll have to change assemblies. We'll move from 38 back to 37, and so we can upload an SRA data set to the assembly. By the way, these demos are based loosely on a set of demos that Wayne, who's sitting across from me, did at ASHG a couple of weeks ago. And so if you want to get that particular handout, which is a nice PDF, the link at the bottom of this slide will take you there. Okay, so I'm going to escape out of my web browser. Okay, so here's the Genome Data Viewer interface here. You can pick a particular organism. We've already selected human for you. If I want to select a different one, I can do that here. I can click to expand the nodes, as I saw on the slide a few minutes ago, to look for a particular organism that's in here. If I want to search the human genome, here's my way to do it. I can browse the genome. I could blast against this assembly if I want. Pick the assembly here, as we discussed. And I can search for lots of different markers that are on the genome or on the assembly. And I'm going to search for adenosine deaminase. Okay, so let's go ahead and run that search. Okay, so here we have the adenosine. We're zoomed into the region surrounding the adenosine deaminase with the default tracks available here. Adenosine deaminase is a gene that's associated with some severe human phenotypes. If there are mutations there, one of them is severe combined immunodeficiency. So let's say we're interested in some kind of clinical features of this particular gene. I can load the clinical features track here by going up here to the tracks menu. And I can go to the NCBI recommended track sets here. So let me load the clinical tracks here. And so we now have the top is our gene map, the adenosine deaminase. Then I've loaded a number of other tracks. This is an interesting one here, and this is the one we're going to focus on a little bit. This is the RefSeq gene alignments to the assembly. And this is interesting because these are particular reagents or records that are designed specifically to give you a stable coordinate system with sort of canonical sequences aligned to them. And it even has the exons spelled out for me and this canonical transcript aligned to it. And so we'll use that sort of as a way of mapping out where we are on the exons of this gene. Down here are all of the variants from dbSNP. Underneath that are variations from ClinVar, our clinical database that talks about the relationship between variations in human phenotypes. And then there are some other kinds of variation tracks here. These are large-scale variants, insertions and duplications and things like that. And then there are other tracks here that have nothing in this particular area. So I'm going to go ahead and delete these tracks, remove them. That's one way we can do to modify the display settings. Now I have a slightly simplified display. Now notice that the track for the RefSeq gene record has an expanded view where I have a gene bar and I have the transcript and protein sequence shown here. Transcripts in blue, the proteins in red. And it might be nice to have this gene view up here for the adenosine deaminase gene sort of rendered in the same way. You can do that by going to the tracks menu here and configuring the tracks. You could also configure the tracks by mousing over the top of the track and using that little gear icon. Or you can use a shortcut, which is this little button here that looks like those tracks. Just go ahead and click that and you will get the various transcripts and protein sequences shown. One thing to notice is that these transcripts are shown sort of with a gray background. And if you go to the help document for this particular track, you'll realize that that's because there are mismatches associated with these compared to the genome. If I mouse over one of these, it will tell me that actually, that there is, this transcript has one substitution compared to this genomic sequence. And that's because the transcript sequences here are based on mRNA sequences that are in GenBank. The genome may have a different variant in it. So notice that there is a little red line here on the RefSeq gene record that basically shows you that it has the mismatch. So it actually matches that messenger RNA sequence. Let's do something interesting here. Let's go ahead and zoom in to exon 2, and I'll zoom to sequence. So we're zoomed in to that region showing that particular change between the RefSeq gene record and the genome. This mismatches at the position of a CT variant, and you can see that variant listed here. And it's actually a common variant. We could add a track to show that. And notice one thing to notice about this gene that makes it a little bit confusing to look at is notice that this basically is written backwards because this is on the negative strand of the genome. I could flip that around if I want to. Let's go to the tracks menu and add another track. So I'll go to configure tracks. So now I have the opportunity to choose from a whole range of tracks here. So let's go to the variation tracks. Some of these are already shown and they're checked here. But one that's not shown on the clinical track are the common variants. So here you can see the ones with global minor allele frequencies of 1% or more. So those would be basically what we would call polymorphisms. And you can see that indeed this variation that is present in this person's genome that was used to assemble this part of the genome, he does have a variant there that's different from, and actually if you go and look at the minor allele frequency, this is actually the minor allele at this position. And this is a synonymous substitution that actually doesn't change that valine codon to a different amino acid. Now notice that there are other variants here, and these color codes here, you can look at that by getting the help document for this particular track. But the purple means that those are pathogenic variants, and if you go to ClinVar, you'll realize that this particular one is associated with severe combined immunodeficiency. Let's go ahead and now we're going to do something a little bit different, something that's not pre-compiled for us. Let's go ahead and add some data ourselves. So I'm going to go to the Your Data widget here on the left-hand side. And I'm going to click this button to add a file. And this is my file. This is from the HapMap individual 12878. And this is a list of HGVS renderings of some SNPs that are in this particular region for this gene. I got this from the Genome in a Bottle data that's available on the NCBI website. And so those variants are showing up for me now in this track at the bottom. And I could have named the track something more informative than the name of the file. And so you can see that person, which is a woman from that Utah population, she has that particular SNP as well. And you can navigate along these if you want to see where they go, where they are. Notice that some of them are present in dbSNP. Some of them, like this one at this particular position in the genome, is not present in dbSNP. So that's if it's not an error, then this is a SNP that's not present in the database. I can use my exon navigator up here to go back to the exon 2 that we were looking at a moment ago. Now suppose I like this and I'm interested in saving this and showing it to somebody else. How could I do that? There's two things you can do. One of them is to go to the Tools menu and to click on this link that says Printer-Friendly PDF. If you do that, you can create a PDF file right here. And I can view that. So that's a nice graphic that I can use to make a figure if I want to. The other option that you have, let me go ahead and cancel this, is to go up here and click on this link that says share this page, and that will give me a URL that I can share with people, and that's good for about 90 days. So let's go on to using some analysis tools from within the browser, and let's move to a different exon. Let me close that. I'm going to go over here to exon 4. Let me center this a little bit better. This is an exon that has some disease gene SNPs in it. You can see the pathogenic SNPs listed here. So suppose we wanted to amplify this, get some primers that would amplify this gene. I can easily do that. If I go over to Tools, I can go to Blast and Primer Search, and I'm going to primer blast the visible range. So that opens up Primer Blast. And if I want to, a reasonable thing for me to do is to adjust these ranges here so that my primers are binding outside of the exon. And I can pick the database. In this case, I want to amplify this from genomic DNA, and it's automatically set for me for Homo sapiens. And if I do that, just to save us some time, I'll show you what the results look like. So we have a set of primers here. I can load those. After I run this, they will be available to me in the graphical sequence viewer. If I go to the Tracks menu here, Configure Tracks, and you'll notice that there's a section or a tab called Primer Blast. And I can just load those. I have several of them here because I've run this a bunch of times. Let me zoom out a little bit so you can see those. And so there they are on the genome. So I have them available and I can see where they would bind and what sequence they would amplify. Let me go ahead and get rid of those. Now suppose I want to do some blast searching against this genome. I can go to a different, I'm going to go ahead and close this Your Data widget up. I can go to a different function or widget here, and that's this blast widget here. I could go to Tools here, run a new blast search. So I have the ability here to do that. I've already set this up with the parameters that I want. I could run these two. This is the mouse adenosine deaminase and the cow adenosine deaminase. I could run those against the genome. I won't do that. I'll go ahead and open this just so you can see what that result looks like. And this file is available on that FTP site if you want that. So here are my results. This is what the mouse alignment looks like to the genome. This is what the cow alignment looks like to the genome. The cow is a little bit better aligned than the mouse is. That blast RID is available over here, and I can go ahead and add it, or if it was a new one or one that I hadn't run in this way, I could just show it by adding the RID. And this device here is a way of navigating across the different hits. There's also a separate larger alignment inspector here. Notice that I have two tracks here. One of them is the blast results themselves. The other one is something that's called the cleaned alignments. Those are a little bit more useful because you can see how the hits from the same sequence are connected to each other. So I could close that one if I don't really want to use it. And so you can use this to navigate across matches based on the messenger RNA sequence here or on the genomic sequence here. So you can see The mouse sequence is the one with the longer accession number, and the cow sequence has the shorter accession number. And typically the cow is a little bit better matched, but not in all of the exons. Okay, let's go ahead and do one more quick thing on chromosome 20. I want to leave chromosome 20 and go to chromosome 2, and we'll do a couple of quick things to show expression and take a look at the last widget down here on the bottom, which is the assembly region details. Okay, so I can jump to chromosome 2 pretty easily simply by going over here, clicking it from the ideogram view, and now I'm on chromosome 2. I can get rid of the alignment inspector 2, and I'm zoomed way the heck out. And I don't want to search for adenosine deaminase. What I want to do is look at a region where there is a mark underneath the ideogram, and that mark tells you that there are some alternate assemblies available for that particular region. There are also maybe some assembly difficulties in that region. You can take a look quick at this one. This is region 116, and it's very near the centromere, which is a region that could be very difficult to assemble. So I'm there, and I need to add a map that's going to give me some information about this particular area. And what I want to do is to use one of my tracks here, and that track is one of the recommended tracks, which will be assembly support. And so you can see there are a number of curation issues in this region. There are some clones that don't match the way that we would like. These are called discordant clones. These are clone-in sequences. And then here is the map that I particularly wanted to focus on. And these are alternate assemblies for this region that represent alternative versions of this part of the genome. If you scroll down a little bit on this genome, you'll see that there's actually a big gap close by. But I can take one of these. One of the things that's kind of cool with this assembly region details is I can take a look. So you can see these marks here. These represent places where there are this particular sequence, this alternative locus, for example, has this alternative assembly, has basically a relative insertion compared to the genome. I can switch this around and change this to the one that is the master sort of in this alignment, and you can see when it reloads, and now you can see the genome has to be gapped to support that one. Okay, let's do the last thing we came to do today, which is to take a look at a different place on chromosome 2, and that's the region surrounding the lactase gene. So here's lactase, and I still have my assembly support tracks up. What I'll do over here is I'll change that. Let's say we're interested in expression. So there is an expression track set available here. And these are a bunch of next-gen RNA-seq runs that are aligned to the genome, and these are used in the process of annotating things so that we can make splice variants and things like that. They also give you a way of looking at expression because these are different tissues or organs or whatever, and you can count the number that align in various ways. Let me return this to the expression tracks. So here is sort of the aggregate of this, but the more interesting thing is to look down at these, which are the individual sort of libraries. So we can see that this gene is expressed in duodenum, as that library is labeled, small intestine. And that's where we'd expect. Lactase is a digestive enzyme. It's predominantly expressed in infants. It is in some cases expressed in adults. We'll come back to that idea in just a moment. Let me zoom out a little bit. And this is kind of a nice example because you can see that lactase has sort of specific expression in that particular organ, whereas the genes on either side are more ubiquitously expressed. Now these are the intron spanning reads. There are other ways of thinking about this. You can just basically look at exon coverages. Where do these RNA-seq runs or reads align to the genome? One way to display that is through the tracks menu here. And I made a set of tracks by going to the configure menu and loading them into the browser and just basically saving them into my NCBI account. So you can definitely save these if you want to. Save the ones you have here. We've already done that for you. But here's one that I saved. And certainly you get similar results. These are no longer spanning the introns, but showing you basically highlighting for you the exons. And we get expression again in this duodenum library. Okay, last trick that I want to do for you today is let's change the assembly and loaded track that we have already that's aligned to this. And I'm going to cheat. There's lots of ways to change the assembly. I could go back to the Genome Data Viewer and switch that there. I think we're going to add the ability to do that to the Genome Data Viewer page directly in a little bit. We haven't got it there yet. What I'm going to actually do is to change the assembly version just by cheating and typing the .25 in there. And you can see that now I'm at GRCH37 patch 13, zoomed out on chromosome 1. I can, just to put us in sort of place we are familiar with, let's find the lactase gene. And now what I want to do is to upload a track. This is a track. It's an interesting one that's available here. It's actually here as one of the supported accession types. This is a SRA run or a next-gen sequencing run for the library RP11, which is used. It comprises about 70% of the genome sequence. But remember, only one allele is included in the reference assembly, so this would be interesting to see what the other allele that this person has could be. So I'll go ahead and load that. And the same kind of thing we saw before, where there are red places, that's where there are mismatches. So there are places where this person is heterozygous for some of the positions in the lactase gene. And we'll do one last search today. And we'll look for the SNP that's responsible for, in some populations, the persistence of lactase activity into adulthood, what provides people the ability to drink milk without gastrointestinal distress in adulthood. And so I'll go ahead and just load that SNP, and we can take a look at this person's genotype at that position. And that's the SNP that I'm talking about. So I'm going to search, instead of with a gene, which we've been doing all day, I'll search with this particular SNP. And it lands me right here, and I'm actually in a different gene. This is in the gene MCM6. All these SNPs are basically landing in the promoter of the lactase gene, so it's upstream, relatively speaking. And you can see that this person is a heterozygote for this change, and he has the allele. The A at this position is the one that allows lactase to be expressed into adulthood. The G, which is the one that's in the genome and is the most common allele worldwide, is the one that's incorporated into this part of chromosome 2. And of course, you can zoom out to see that this is near the, if I change the region here, let me just go to gene with Pat to zoom out a little bit faster. And I'll just slide it over a little bit. And this is lactase over here. And upstream of it is MCM6. And so that's a lot that I did today, I think. Maybe that was a little bit too much. We were a little bit over time. But let's go ahead and pause now, and we'll answer any questions that we can. And if we don't have any questions, we'll take a couple of minutes to let people think if they have any. And I'll just share this slide with you at the end here. There's a number of useful links on this slide for the Genome Data Viewer. There is a help document. There is a YouTube video that Wayne made that's available now in the public, basically just an overview of the Genome Data Viewer. There is a fact sheet available on our FTP site. In general, please check out our YouTube channel. We make videos of all these webinars. We also put instructional videos up there. There is a Learn page at NCBI, which you can get to from the home page. And that URL there will take you to the fact sheets directory. There are fact sheets on just about any NCBI resource you can think of. And if you have questions, check out our support center. There are a lot of answers to questions there, and you can write to the help desk from that page. So Wayne doesn't see any questions there. If you have questions about the webinars in general, you can write to us at webinars@ncbi.nlm.nih.gov. If you have any questions in general about NCBI, you can use that support center link that was on the previous slide, or you can write to info@ncbi.nlm.nih.gov. And of course, you can always write to me, peter.cooper@nih.gov. Okay, thanks everybody for coming. Okay, I'm going to go ahead and end the webinar. Thank you.